actually a pretty major mistake on the problem set. This is pertaining to problem 4A, where you have to draw the structure of an alpha keto acid that results when an amino acid undergoes deamination through a transamination reaction. So remember that in transamination reaction, you have an amino acid. So amino acid, recall you had your alpha carbon. So here's alanine, you have an alpha carbon attached to amino group, side chain, and carboxyl group. In the transamination reaction, you have removal of the amino group and a proton at the alpha carbon, and that's replaced with a C double bond O. So what is wrong with the structure that I've drawn? Yeah, right? There's an extra bond attached to a hydrogen, so that causes there to be 5 bonds attached to carbon, which is like the #1 rule to never break in chemistry. So that is what's wrong with it. And this is actually wrong for each of your structures. So so your product should really be the alpha carbon attached to C double bond O attached to carboxyl, and then your side chain. And you should not have that alpha proton there, and the amino group should be removed. And that's the same for alanine, leucine, as well as phenylalanine. So it's a good review of the quiz that we have at the end of the hour on amino acid metabolism. A few other announcements. Next Tuesday, the literature assignment is due. Remember that if you wish to, you can work with a partner and hand in one document of the answers, or you can work by yourself. And then next Thursday is exam 3 already. So things are just really moving along in this class. I know. Even I can't believe it. I'm just like, what? There's another exam? So I know how you guys must feel. All right. So we left off last Thursday, I believe, talking about the structure of nucleotides, specifically deoxynucleotides, and now, as well as DNA, and now we're going to go into sort of the structure and function of RNA, and then go into nucleotide metabolism. It would help if I had this plugged in. Okay, so now we're going to talk about RNA. Now I realize that most of you already know a lot about DNA and RNA because you probably heard it in your other classes such as Bio 120 or Cell and Molecular Biology. So if you already know this information, then this is great review for you, and you can kind of sit back and just enjoy listening to a review of the information. If you don't, then obviously we're going over new material. Now, one thing I do like to do, because this is a chemistry class, is I really do like to stress the chemistry of RNA and DNA and genetics. And I'll definitely sort of let you know when I think there's something in terms of the chemistry that's really important for understanding for, say, on exam. Okay, so RNA is the second major form of nucleic acids in cells, and RNA has lots of different functions. In terms of gene expression, RNA is going to act as the intermediary molecule between DNA and protein. So it's going to use the information encoded in DNA to specify the amino acid sequence of a functional protein. So in eukaryotes, DNA is confined largely to the nucleus. And we know that in the cell, protein synthesis has to occur on ribosomes in the cytoplasm. And so there has to be some other molecule that carries the information in DNA from the nucleus into the cytoplasm in order for translation to occur, and that is RNA. And so in the 1950s, RNA was considered kind of this logical candidate that connects the genetic information in DNA and proteins. And this is because it's both found in the nucleus and cytoplasm. And it increases protein synthesis by, if you increase RNA in the cytoplasm, then you'll see an increase in protein synthesis and an increase in the rate of turnover. Okay, and then in addition to sort of this information carrying role of RNA that RNA has, there's other RNAs that play different structural, regulatory, and even catalytic or enzymatic roles. So this table lists kind of the major different kinds of RNA that are found in the eukaryotic cell. So there's transfer RNA. These are relatively small RNA pieces of RNA, and they're going to be important for attaching the RNA to amino acids and being involved in protein synthesis. There's ribosomal RNA. There's different kinds. They vary in size. They are going to combine with ribosomal protein, and they are going to be the site of protein synthesis. There's messenger RNA. These come in variable sizes. This is probably the RNA that you are most familiar with, also known as mRNA, and it's going to direct the sequence of amino acids and proteins. And then there's sort of these smaller RNAs that have kind of more regulatory functions that we'll talk about later in the course. So there's small nuclear RNAs. They're involved in the processing and maturation of messenger RNA in eukaryotes. There's small interfering RNAs, also known as siRNAs. So they are involved in regulating gene expression. They tend to essentially, they're involved in what we call gene knockouts, or lowering the levels of an expressed gene in a cell. And then there's also microRNAs. These are also small RNAs that are also involved in gene expression. Okay, so in terms of RNA structure, for the most part at least, mRNA, RNA is largely single-stranded, and it can form a single helix. But RNA, if it has the right sequence in the actual strand, it can self-base pair in the strand and form a double helix. So when it forms a double helix, it tends to form sort of that A form of the helix. So that's kind of that more condensed form that we see, as well as other structures. So for example, you can have an A-form helix, you can have loops form or bulges, depending on if you have atypical or aconical or non-conical base pairing of the bases with each other in the RNA sequence. Or you can have even hairpin structures if you have complementary nucleotide sequences that are interspaced with non-complementary sequences. Okay, and so RNA often contains, like I said, non-canonical base pairing. Example, G base pairing with U, and that's going to contribute to the overall three-dimensional structure. So we're going to go, so transfer RNAs, again, these are smaller RNAs. They're approximately 80 bases. Each at the 3' end of the transfer RNA sequence, there is an amino acid that's going to covalently bond to the 3' end. And so the transfer RNA is going to be important because it's going to bring the amino acid to the ribosome for translation. And transfer RNAs also tend to have non-standard bases, and they are generated by post-transcriptional processing of these non-standard bases. So these are some of the non-standard bases that are in transfer RNA. terms of the structure, you can have, again, this interchain base pairing, and this interchain base pairing can cause these secondary structural elements called hairpins, or also known as stem loops. So remember that hairpins or stem loops are really brought about when you have, within those same piece of DNA, two regions of sequences that are complementary, that are basically interrupted by non-complementary sequences. And so the complementary sequences can base pair with each other, and then the non-complementary sequence forms kind of this bulb. And that's what we call the stem loop or hairpin structure. And so the transfer RNA has essentially three of these little stem loop or hairpin structure in kind of its overall three-dimensional structure. And so this is sort of actually a closer representation of the three-dimensional structure of transfer RNAs. So it forms this sort of clover leaf structure, and this is really the full three-dimensional structure where you have hydrogen bonding from base pairing as well as base stacking interactions. And you need to have this full three-dimensional structure for activities. At the bottom of the tRNA is what we call the anticodon. So this is going to sort of be important for coding information in mRNA. And then at the 3' end of the tRNA is where the amino acid is attached. And when we go into translation, we'll talk about how the mechanism of attaching the amino acid onto the tRNA molecule. Okay, ribosomal RNAs, they're large. There are only a few types in prokaryotes and eukaryotes. Ribosomal RNA actually make up 60 to 65% of the ribosome. So the ribosome is that huge nucleoprotein machinery that's involved in translation, and it's actually made more RNA than there is protein. So this is sort of the two different, what we call subunits of the ribosome, and in salmon, in yellow, that represents, it should really be DNA, or sorry, RNA, not DNA, and then the purple represents protein. Magnesium ion is really important for sort of keeping the structure of the ribosome together, and the RNA in the ribosome is going to be important for binding to the mRNA. Ribosomal RNA is really classified by this, what we call the sedimentation coefficient, which the sedimentation coefficient is really kind of this unit of, it's kind of a unit for molecular size and shape. And so when people initially isolated the ribosomal RNA, they would essentially centrifuge, and the ribosomal RNA was centrifuged at this specific size, which we call the sedimentation coefficient. So S represents basically these different parts of the ribosomal RNA, and they each have their own size and shape. And so actually, this is a prokaryotic ribosome. It has what is kind of in its big complex. It forms the 70S shape, so it's a bigger shape and size. If you were to remove magnesium from the complex will dissociate into two different subunits. This is called the 30S and 50S subunits that are smaller than the 70S. And so that's why we call them, these different subunits, because they have different sedimentation coefficients, or essentially different sizes and shapes. Okay, so our RNA structure is actually really quite interesting and very complex. rRNA really forms very complex, intricate structures that are characterized by lots of different stem loops, bulges, non-canonical base pairing. So this is the tertiary structure of RNA. And then if you can imagine this RNA being incorporated into the ribosome, so this is the ribosome, the 70S ribosome, that includes both the protein and the RNA intermingled with each other to make the functional ribosome. This is what it looks like. So kind of the purple little strands, those are, that's protein in the ribosome, and then everything else, so this blue and gray and yellow, that represents the rRNA part of the ribosome. Yes, Alex?
Is there a functional purpose to the hairpin structures and the loops, or is it just that's just the way it is?
Well, there is. It's forming its overall tertiary structure, three-dimensional structure. So yeah. So and then forming kind of the overall three-dimensional structure of the ribosome is what gives it its function. So great question. Yeah, I mean it's definitely not random, right?
Right.
Yeah. So, and this is sort of drawn to be. So this schematic, what this is really showing is the base pairing interactions between the different bases in the RNA molecule. So it's kind of a linear representation, and then that linear sort of molecule is going to sort of fold up into this very more complex, structure that gives the ribosome its shape and function. Did you have a question, or are you just stretching?
No.
Oh, you're just stretching? Yeah. Okay, so we're just going to zoom in to sort of this little region of the ribosomal RNA. And so this kind of just zooms in and shows the base pairing interaction of the RNA molecule, the interchain base pairing of the RNA molecule. So you have your sort of canonical Watson-Crick base pairing in the RNA molecule where, G's base pairs with C's, A's base pairs with U's, right? So in RNA we have uracil, not thymine. But then you have these non-canonical interactions such as U base pairing with G as well. Do you have a question, Mitch?
Yeah.
Yeah.
So, why is ribosome structure, our RNA structure, why? So, these things that are able to be done with the ribosome or the RNA.
Mm-hmm.
Why can't that be done in DNA? So, for example, the loops or it doesn't tend to happen. Am I wrong on that?
Yeah, it doesn't tend to happen, no. And I think that's more to do with, that's a great question. Now, it tends not to happen in DNA so much. And I think that's, well, there are certainly parts of DNA that have unusual structures, okay? And that's, when we kind of talked last week about DNA forming a triple helix or a quadruple helix, and that has more regulatory function. I think that it really goes back to the fact that DNA is important. It's the genetic material. It's the actual sequence of the A's, G's, C's, and T's code for protein, okay? So it has sort of this, sort of a more canonical helical structure for sort of keeping that information stabilized and protected. Here in RNA, we have kind of these different structures that form, and it's because these different little loops and stems are actually contributing to an overall three-dimensional structure that has a specific function. Does that make sense?
Yeah.
Yeah, so tRNAs has this the three-dimensional structures having this specific function of interacting between the RNA and the ribosome. The rRNA, it's actually it's folding into it's almost like a protein in a way. It's folding into a three-dimensional structure in the ribosome. That's my thought. Do you have a thought on it?
Could it be also because RNA is single-stranded and like it doesn't have another set of strands like complementary?
Yeah, so you have, yeah, so actually that's probably a great point, is really just sort of fundamentally because RNA is single-stranded. So it does base pair with itself. So it does kind of form, it's going to have different properties compared to DNA that's double-stranded. So, but it makes these very intricate, complex structures, which are, it's pretty interesting. Great questions. I love it. Okay, so messenger RNA, this is kind of like, this is probably the RNA that you know the most about. It turns out, so this is really the RNA that codes for, right, genes, which codes for proteins. And interestingly, a messenger RNA is actually the least abundant form of RNA in the cell. It's primarily single-stranded, and so mRNA is going to be, it's that unit that essentially transfers the genetic material from the nucleus into the cytoplasm and code for protein, and then code for protein. So mRNA is transcribed from DNA, so it's essentially sort of this temporary copy of DNA, and then once it's transcribed, it is then shuttled out of the nucleus into the cytoplasm. It then interacts with the ribosomal machinery and tRNAs and translated into protein. Messenger RNA tends to be rapidly degraded, so there's very fast turnover of messenger RNA. Why do you guys think that could be? Why is messenger RNA rapidly degraded?
Because of the exon and intron?
You know, that could certainly be part of it. I mean, certainly there are enzymes that are going to, you know, excise exons and introns. So certainly messenger RNA is very accessible to enzymes. Stephanie?
Isn't it highly unstable?
It is. So why is it highly unstable? Any other guesses? William?
It's because its bases are exposed to the cytoplasm.
Right, it's single-stranded. Yeah, it's single-stranded, right? So double-stranded DNA is much more stable because it's double-stranded and it's just a very stable helical structure. Okay, so messenger RNA is very heterogeneous in size and sequence because it's really coding for, it's a copy of a gene, you know, so it's a copy of a genetic material, and each gene is of different size. We know that, right, messenger RNA contains exons. That's the coding region that is going to be translated into protein. It also contains non-coding regions called introns. They will be spliced or removed prior to translation. They will also receive what's called a 5' cap as well as 3' poly A tail. This is really important for stabilizing the messenger RNA product and important for regulating translation and splicing. And then it has codons where in the coding region, each three sequence of bases is going to specify for amino acids and or specify the amino acid sequence of proteins. Okay, so prokaryotic mRNA. Well, first let's talk about a cistron. So a cistron is a gene, and a single mRNA molecule can be monocistronic, meaning that it carries the code for only one polypeptide protein chain. Okay, so this represents a monocistronic piece of mRNA that contains that genetic information that only codes for one protein. You can also have what's called polycistronic mRNAs, and that's where you can have where it can encode for two or more different polypeptide or protein. So you could have kind of within the same piece, same mRNA molecule, the coding sequence of gene 1, gene 2, and gene 3. And so this is kind of a property of prokaryotic RNAs or mRNAs. They tend to be polycistronic, where you can have one piece of mRNA that can encode for multiple genes, and they often are involved in the same pathway. So it's sort of coordinating the regulation of one entire pathway. Okay, eukaryotic, the mRNAs tend to be monocistronic, so one mRNA is going to encode for one polypeptide product. So the minimal length of a mRNA sequence is really set by the polypeptide chain for which it includes. So remember that 3 nucleotides, it's going to encode for 1 amino acid. And so if a polypeptide contains 100 amino acids, then it has to minimally have 300 nucleotides in the RNA sequence. But usually it's much longer. So the mRNAs that are transcribed from DNAs are somewhat larger in length. And that's because you have sort of these additional sequences, so sort of the non-coding RNA sequences such as introns, as well as regulatory sequences at the 3' and 5' region. Okay, so that kind of concludes sort of like our discussion on mRNA, and now we're going to go into the sort of the annealing and melting properties of of RNA and DNA. Okay, so DNA, this is really more specific to DNA than RNA, although it can happen with RNA too. DNA can undergo reversible denaturation, that is melting, and annealing or renaturation. So you can actually disrupt the hydrogen bonding of the bases of DNA by that's denaturing it by changing temperature, so making it really, it's really actually heating it to very high temperatures or changing the pH. Mitch?
Can you just like use like helicase?
You could certainly use an enzyme to disrupt the base pairing of DNA. Yes, that's going to be a little bit more specific to like what goes on in the cell. But if you were wanting to do this in an experiment, you could just use heat to essentially take a double-stranded DNA, disrupt the hydrogen bonding interactions, and then make it single-stranded. Okay, so you can disrupt the hydrogen bonding between the double-stranded DNA molecule, so the hydrogen bonding of the base pair as well as the base stacking interactions by applying heat or changing the pH. That will unwind the double helix and make it single-stranded, so the 2 strands of DNA are completely separated from each other. So this doesn't break covalent bonds. Okay, so you don't break any phosphodiester bonds within the backbone of the DNA. It's just really breaking the non-covalent bonding interaction. And you can actually renature, that is, cause the DNA to reform a double helix by cooling down the DNA. So it's going to actually form the original double-stranded helix. And this property of being able to reversibly denature and renature DNA is the basis of many techniques that are widely used today, such as PCR, microarrays, and southern blots. All pieces of DNA can have what's called a melting temperature. So when DNA is denatured, so you have your double-stranded DNA, you apply heat to it, the strands separate, and then the bases unstack, okay? And then the bases, they interact with water. So you have sort of the aromatic bases interacting with water, and this actually causes a change in the absorbance at 260 nanometers, okay? And this is called hyperchromicity. And this property is important because you can use that change in absorbance to determine the melting temperature of DNA. So Tm is the temperature where 50% of the DNA has melted. Okay, that is, it's formed single-stranded DNA. And you can actually, measure that by measuring the absorbance at 260 nanometers. Okay, so if you have a higher percentages of GC, that's going to lead to a higher melting temperature. And this is because more energy is required to break the hydrogen bonds. Why is that? Why would higher GC content lead to higher melting temperatures? Yeah, because there's 3 hydrogen bonds compared to AT that has 2 hydrogen bonds. Okay, so this plot right here is looking at the percent denaturation as a function of temperature. And looking at this plot, what is the melting temperatures of the DNA in this graph? Based on the definition of Tm. What do you guys think? What was that? Yeah, well, about, okay, so let's look at, so Tm is a temperature whereby 50% of the DNA has denatured. So if we look here on our x-axis, this is the percent denaturation. So this is about 50%. We're going to go across and down, and for the blue strand, the Tm is going to be about maybe 82.5, right? Or else the red strand, if we go across at 50% and go down, that Tm is approximately 85 degrees, so 80 and 85 degrees. So then which one has the higher GC content? The red one, right? Because it has a higher melting temperature, which means there's more energy required to separate the 2 strands, and that's because there's more hydrogen bonding interactions due to the greater GC content of the DNA. Okay.
Can you explain the bottom graph? Like, what would you use that for? Because I thought your question was towards the bottom graph, and I was like,
Oh, so this bottom graph here is just showing that there is a relationship between the melting temperature and the GC content. So that is the higher the GC content, the greater melting temperature. So I would probably ask you a question based on this type of information up here in this upper graph. Okay. So nucleotides and nucleic acids can undergo non-enzymatic transformations. So they can have spontaneous alterations in their structure. The rates by which these spontaneous alterations occur, they tend to be very slow, but they actually are very physiologically significant because the cell has very low tolerance in alteration of genetic information. And so an alteration of genetic information is a mutation, and this is where it causes a permanent change in the information that's encoded in the DNA. And there's there's very, very good support to suggest that there is a very intimate link between the accumulation of mutations in your DNA to the process of aging as well as carcinogenesis in organisms. So it's really thought that, well, I would say this is a little controversial, but it's really thought that the process of aging is, it's certainly linked to your DNA acquiring more and more mutations over time, which also contributes to cancer development. However, we know cancer development is, right, it's much more complicated than that, right? Because we learned that metabolism has an important role in cancer development. Okay, so here's just an example of mutations. You can have deaminations. So for example, cytosines cytosine bases, they can undergo deaminations where the amino group is replaced with a double bonded oxygen to make uracil. That can happen with 5-methylcytosine to make thymine, as well as adenine and guanine can be deaminated to make these sort of intermediates called hypoxanthine and xanthine. You can have dimers that can form between two adjacent thymine bases. So if you have two adjacent T's next to each other and it's exposed to UV light, for example, when you're in the sun, right, then the two thymines can form covalent bonds with each other. So that can produce a dimer. You can also have what's called depurination, where purines can, a purine nucleotide or a purine within the DNA can lose the base, resulting in an apurinic residue.
Question.
Yes.
So for like intercalators,
Mhm.
bromide or thinning bromide, things like that, how do those like cause mutations? Like supposedly they can.
Mm-hmm.
How would that?
You know, I don't exactly entirely know how it actually causes mutations. I don't know. That's a great question, because I know it can, and I know we certainly wouldn't want to spill a bunch of it on our skin. But yeah, that's a great question. Do you want to find out for us? You have a very long list. Okay. You know what? Let's see. We went through a lot of lecture material. Why don't we do a little problem solving that's going to relate to this material here. Let's do some problem solving based on Irwin-Chargraff's rules. So remember, Irwin-Chargraff came up with these rules that in DNA, the percent A equals percent T, percent G equals percent C, and then A plus G equals T plus C. Okay, so let's say that you had a piece of DNA and the percent, I'm really just throwing these numbers out there. The percent A equals 20%. Let's do actually two different pieces of DNA. Okay, so DNA A has a percent A of 20%, and then DNA B has percent A of, let's do 30%. Okay, so what are the percents of T, C, and G, T, C, T, G, and C in each nucleotide sequence? Okay, so why don't you guys take a moment to figure this out? I'll give you a couple of minutes. Do you have a question, William? Is it William or Bill? Okay. All right, so why don't you guys take like 2 minutes and figure this out. And then also, which one is going to have the higher TM? And I would probably ask a question like this on an exam too. Just throwing it out there. just give you guys about 50 more seconds and we'll go over the answer. All right, so we're just gonna go ahead and go over the answers. Okay, so we're given information where the percent A is 20%. What information can we use to determine the percent T? Yeah, right, percent A equals percent T. So we can already figure out percent T is what? 20%. And then percent for molecule A, for molecule B, percent T equals 30%. So then how do we figure out percent C and G? What was that? Yeah, you can use the third equation. Alternatively, right, this really has to add up to 100%, right? So that means that if so 100% minus 40%, that equals 60%. So 60% of the DNA has to be G and C. And we know that percent G and C are equal, so therefore G is what percent? 30%. And C equals 30%. And then by that logic, then we can assume that a similar type of, we're going to do a similar type of rationale for solving this problem for molecule B. So we know that A and T equals 60%. That means G and C is 40%. And so G should equal 20% and C equals 20%. And then which of them is going to have a higher melting temperature? A. Why is that? Yeah, because it's got the higher DC content because? Yeah, more hydrogen bonding, right? Okay. All right. So just a little refreshing break, right? Now we're going to go into part 2 of nucleotides, which is their metabolism. So I'll do maybe about 15 to 20 minutes of this. Yes.
So I googled the affinity bromide.
Uh-huh.
It's because it inserts between base pairs and cross relationships.
Oh, that's interesting. Okay. Thank you. So you have one less thing to report on. Okay. All right. So we're going to go to talk about nucleotide metabolism. There's a lot of reactions in nucleotide metabolism. I will let you know what it is that I want you to actually learn. There's a good chance that we'll get to so far and we're not even going to cover all of this. Okay, so I'll let you know what it is you'll have to be responsible for. It'll be very clear based on what kind of answers I ask in the problem set. Okay, so remember, in terms of nucleotides, you have your purines, so that's going to be your bases that have the 6-member ring attached to the 5-member ring, and that's adenine and guanine. And then your pyrimidines are your 6-membered ring bases, so that's cytosine, thymine, that's in DNA, and then uracil, which is in RNA. And so purine and pyrimidine synthesis are going to be, they're going to be synthesized through different mechanisms. Okay, so there's essentially 2 types of pathways by which nucleotides are made. And there's what's called the de novo pathway, meaning they are made from new, and then the salvage pathway, which is essentially the recycling of already existing nucleotides. So de novo synthesis is going to be derived from different metabolic precursors such as amino acids, ribose 5-phosphate, CO2, and ammonia. And again, the mechanisms for making purines and pyrimidines through de novo synthesis pathways are going to be distinct. The salvage pathway is going to essentially be taking some of these bases and recycling them. So we're going to first go over the biosynthesis of purine nucleotides. And first, we want to talk about where it is that the ribose part of purines come from. So in de novo synthesis of purines, the base is actually synthesized directly on the ribose backbone. Okay, so the ribose is going to act as a carrier of the nucleotide in the synthesis. This is going to be distinct from pyrimidine synthesis, which you will want to know. So ribose 5, so remember ribose 5-phosphate, okay? Where does ribose 5-phosphate come from? You guys remember that molecule? Pentose phosphate pathway, that's right. Remember, ribose 5-phosphate was one of the products of the pentose phosphate pathway. What else was a product of the pentose phosphate pathway? An ADPH, yes, great. Okay, so remember ribose 5-phosphate is a product of the pentose phosphate pathway. It's made under the fed state in response to lots of glucose being around in the body. If there's lots of glucose around in the body and you have lots of ribose 5-phosphate, then that's a signal to the cell to say, hey, lots of nutrients around. Why don't we grow and replicate and divide? So we're going to need more nucleotides. Okay, so we have ribose 5-phosphate, and ribose 5-phosphate is further phosphorylated, so ATP is going to be the donor into this key intermediate called phosphoribosyl pyrophosphate, or PRPP. So PRPP is essentially going to start as the sort of the starting material or the backbone upon which the base of the purine is going to be built upon. So the first thing that happens is we take our PRPP molecule and it's reacted with a glutamine. Okay, so glutamine is going to be attached to carbon 1 of PRPP, and then that's going to release pyrophosphate to then make this intermediate called 5-phosphorybosylamine. Okay, so we have this 5-phosphorybosylamine. Here's your amino group attached to carbon 1 of the glutamine, and that is going to be sort of the point where the purine ring is going to be built up from in the structure. My computer is frozen. Okay, and this reaction is important because it's the first committed step of the pathway. What do we know about committed steps in pathways? Yeah, they're irreversible. They tend to be irreversible. Anything else? Yes, they tend to be regulated, right? So any step that's committed tends to be regulated, and it's also irreversible because the energy of that step tends to be very favorable. Okay. in terms of that reaction. Okay, so so the second step of the reaction is a condensation reaction in which glycine is attached to the amino group through a condensation reaction. So the carboxyl group of glycine is phosphorylated, making an acyl phosphate, which activates the glycine molecule for then attaching that carboxyl group of glycine onto the amino group, forming this intermediate. In the next step, a formyl group, which is kind of this aldehyde group, is attached at this. So a formyl group is attached at the amino group of the glycinamide ribonucleotide intermediate, and that formyl group is actually coming from this this molecule called N10 formyl tetrahydrofolate. So this is the structure of N10 tetrahydrofolate, and the formyl group is essentially being transferred onto the amino group to make this intermediate. In the next reaction, we have addition of another amino group. So this is going to be coming from glutamine. So at this carbon here, an amino group is going to be attached to glutamine. In the next step, we essentially have all of the molecules that we need to make the 5-membered ring of the purine. So in the next step, we have a dehydration reaction in which a molecule of water is removed, and then we have ring closure between this nitrogen and carbon. So it forms a bond and yields the 5-membered ring. Okay, so at this point, we have 3 of the 6 atoms needed for the second ring in the purine structure. So it's these 3 atoms, this carbon, and this nitrogen. And in the next reaction, a carboxyl group is attached to this nitrogen using bicarbonate as the carbon donor. And so this reaction is kind of unusual because it requires no biotin. So once we have carboxylation, then there's a rearrangement reaction in which the carboxyl group is transferred from this nitrogen to this carbon. And then in the next 2 steps, we actually have attachment of the amino group, and that's coming from aspartic acid. So aspartate is attached to this carboxyl group through a condensation reaction. Or it's attached and it forms an amide bond through a condensation reaction, making this intermediate. And then the next step is the carbons from aspartate, they're actually going to leave as fumarate, and it's going to leave that nitrogen behind. So it's going to leave behind that amino group. Does this look like anything you guys have seen before, where you have an amino group being attached, aspartic acid being attached to a molecule, okay, and then fumarate, the carbon skeleton leaving the amino group behind. Yeah, the urea cycle. So if you guys remember, in the urea cycle, you have citrulline. The citrulline is going to react with aspartate, making arginosuccinate, and then fumarate is going to leave. So that's the carbon skeleton of aspartate leaving that amino group behind, forming arginine. So this is just kind of a common theme that is both present in the urea cycle as well as nucleotide biosynthesis. Okay, and so in the next reaction, we have another dehydration reaction where a molecule of water is removed, and then we have ring closure, making the completely closed purine ring. This makes an intermediate called IMP, so inosonate monophosphate. And then IMP is going to essentially, its synthesis is going to diverge to make the two different purine molecules. So this slide is just really an overview of what we went over so far, starting with PRPP being made into IMP. And so once we have IMP, then IMP can be used to make GMP or AMP. So for IMP, or to make AMP, it starts with IMP. It's reactive with aspartate to, so aspartate is essentially condensed onto IMP to make this molecule. And then fumarate is going to leaving behind that amino group on the molecule to make AMP. For the synthesis of GMP, you take IMP. IMP first becomes hydroxylated at this carbon, and then a hydroxyl group becomes oxidized by IMP dehydrogenase to make XMP. And then XMP is then amidated, where the amino group, where it's reacted with a glutamine to donate that amino group to then make GMP. Okay, so in terms of this sort of the structure of purines, you should know where each atom of each of the purine molecule is coming from. So you should know that some atoms, so nitrogens are really coming from aspartic acid, glutamines, this part is coming from glycine, and the carbons are coming from CO2 and formic acid. Okay, so there's a lot of regulation of purine biosynthesis. We will go over regulation when we come back on Thursday. But now, I think you guys know where we're heading, right? Put everything away, it's time for the quiz.